thank you, Yvonne, for giving us an introduction that was actually good. So setting the stage for what we think about in the context of immune therapy. So today I'm going to talk about some of the things that we're doing in the lab. And of course, this is a shortened version of all the things that we're doing. So it may be a little bit disjointed, so please, ask a question afterwards. So we do things a little bit differently. Part of our story is developing antigens, as Yvonne said, so antigen discovery, but we discover things in a particular way. We're interested in carbohydrates that are expressed on tumor cells. And as I'll show you, there's tons of carbohydrates. And you can make carbohydrate vaccines and do all the things that people have been doing. And most of our successful vaccines in bacteria are carbohydrates, so why not for tumors? So certainly there's a lot of effort. But when you start to think about those carbohydrates, you start to think of them in the context of multivalent or polyvalent type vaccines. We came back almost 30 years ago now, it seems like, longer than I want to think about, and said maybe we can take a different approach, and that is basically developing peptides that mimic carbohydrate. And that does a couple of different things for us. It allows us to think about T cells and B cells, but if you can reduce everything down to one peptide, maybe you can get a shotgun approach, okay, to hitting all carbohydrates, not just, so it basically defeats the purpose of developing multivalent and all the issues that come with that. Maybe you can get the same thing with one peptide. So I want to talk a little bit about that. So we've talked about, in the context of the foundation of our story, the rationale. If we're going to use one peptide, we're still wrestling with this concept of a pan-specific response. Bacterial people talk about this. If you say I have 5 or 6 different serotypes and I'm targeting a core antigen on those 5 or 6 serotypes, I'm developing an antibody that sees that core. We're saying something a little bit different than that. So I'm not quite sure the pan-specific response is the right term, but we're still wrestling with that. So I'll talk a little bit about the discovery phase. And we've moved this peptide, a peptide, into phase 1 and to phase 2. And I'll talk a little bit about the phase 1 trial, just snippets of that, and a snippet of what we're seeing so far in our phase 2 and what the expectations are. Okay, so again, as I was saying, carbohydrates generate B cell responses. They really don't, even if you conjugate them to carrier proteins, they're really not carbohydrate-specific responses, cellular responses. So we came back and said, well, maybe we can circumvent this cooperation by making peptides that link B and T cell epitopes so that we get a humoral response with an anti-tumor cell response. And over the years, we've published 60 papers on how this works. But in the context of how we think about these things a little bit differently, we think of each CMP as a pan-antigen, meaning that and I'll show you a little bit about what that means, but thinking of one peptide that interacts with a variety of different antibodies that see different carbohydrates or with lectins. The idea is to generate a polyspecific response. We do generate anti-tumor T cells. We've published on that, but I won't be talking about that today. The beauty of peptides, though, is that you can, unlike carbohydrates, you can use multiple platforms. You can stick them into listeria. You can stick them into DNA plasmid. You can do all these things that you can't do with a carbohydrate. And more importantly, because of the nature of the cellular response we generate, and we've shown you can generate long-term memory responses, which you cannot do with carbohydrates. Okay, so what we really want to understand here is how to harness the inherent polyspecificity of antibodies to target multiple tumor-associated carbohydrate antigens. This is what innate immunity does, right? When a bacteria becomes infected, you become infected with bacteria, the first line of defense are antibodies. And they see all kinds of things. Why can't we harness that concept to look at a lot of different carbohydrate forms? Because carbohydrates, they're either glycolipids, glycoproteins, they impact the biology of a cell, and they also are involved in cell death. So if you think about polyspecificity, it has the potential to target multiple carbohydrates as opposed to making a polyvalent vaccine that has GM3, GD3, GD2, Lewis Y, blah, 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 right? So it has the potential to synergize also with other therapeutics to enhance cell death if you are able to generate antibody responses that somehow impact in the context of cell death signaling or apoptosis, and that's really what we've accomplished. So the way we went about this is thinking, well, we have typical phage display library. And this is what generally happens. But people usually have one monoclonal, and they choose one peptide. And over the years, we showed that you really can't do that. There's a lot of other immunological concepts. Where does the peptide bind? Is it only the heavy chain, only the light chain? Do you have to have heavy and light chain? The other part of what we've shown is that you can take a peptide, and these are the two peptides I'll be talking about today, P10 and P10 short. Those two peptides react with two types of monoclonals that are involved, BR55-2, ME361. They see different antigens. Those peptides cross-react with those two monoclonals. Those two monoclonals trigger apoptosis. They trigger signaling. So what we want to do is develop an immunogen that induces antibodies that do exactly the same thing as these two antibodies, or with lectins. So the process we thought about is, do you have binding, lectins, antibodies? We have peptide selection. We've gone through the trouble of remodeling things, and I won't talk about that today, using crystallography, NMR analysis, and molecular modeling to develop these two types of peptides. You immunize with these peptides in mice, and we've shown that you can follow the circle and come back and actually develop antibodies that induce apoptosis, and they bind to a lot of different carbohydrate forms. Just in passing, with Dorothy Hurley at the Wistar, we had shown in a program project that we have with her that this P10 peptide, through the process of random selection on these phages, certainly inhibits tumor cell growth. So we know we have a starting point with a peptide that does what we want it to do. But more importantly, we want to know what's happening with human immune system, right? I don't care about the mouse. I want to know something about the human system. So we decided to make some columns, and there's a conception here in terms of innate immunity. Do you have circulating carbohydrate reactive antibodies, okay, that actually do something? So are they part of immune surveillance for a tumor cell? So we made columns of these two peptides, passed over IVIG. And the most important thing here is the type of isotype that was enhanced on these columns, and they were IgG2. These are typically carbohydrate reactive antibodies. So we know we are now able to isolate human antibodies. And the question is, do they do something? And they're carbohydrates. So again, the mimicry component of this, the peptides are mimicking true carbohydrates. The other part of those antibodies is asking what else do they bind to. We sent off the antibodies to the consortium to come back and say what other types of carbohydrates do they see. They see GD2, GM3, fucolac, lac, all the things that we were interested in expanding. So the question is, if I immunize with these peptides, do we get that type of response? So we know we can isolate these human antibodies, but if we immunize with this, do we get the same sort of responses? And that's what we've shown here. So we had published a paper in 2007 that if you take a shorter version, this P10S, and immunize relative to P10, you get enhancement of GM2, GD3, GD2. So P10S was designed as a more faithful mimic of these carbohydrates. And if it's a more faithful mimic, you should be able to induce an immune response that's more likely to cross-react with the types of carbohydrates you're interested in. And what's also interesting that we found was that the induction of those antibodies correlated with the antibodies that we isolated from human IVIG. And it's pretty much a straight line with a 0.82 R factor. So we can induce and isolate very similar antibodies. And those antibodies do something, right? So we showed that this is a MDA Met cell is a bone colonizing cell. It's a breast cell that was derived from MDA 231 cells through a variety of intercardiac immunizations, which we had published on a long time ago. But in fact, if you take those antibodies that we've isolated on P10 short, you can block the migratory properties of MDA Met cells. So they actually do something for us. So we said, okay, fine. We like this peptide. Let's move it into the clinic. We had gotten funding from the DOD for breast program. We did all our GLP studies were done at the University of Arkansas. I'm probably more proud of the IND application. It's over 1,600 pages. And we only ended up with 6 questions from the FDA. So we were spending a lot of time just agonizing over this. The types of adjuvant that we used was montanide. We had used QS21 in the preclinical studies, but because of indemnity issues, all of a sudden it popped up. We had to switch adjuvants. The peptide itself is P10 short, but we also synthesized it with PADRE, the universal T-cell epitope. So in mice, we saw no adverse responses. We sent everything off to the FDA, and of course, they said, fine, let's move ahead. So our phase 1 was a classic 3+3. And the reason we chose this classic 3+3 is really because I was kind of concerned about the induction of cytotoxic antibodies. 3+3 designs are kind of out of vogue right now for cancer vaccines, but most of the cytotoxic drugs that were developed over the years used the classic 3+3. So I basically erred on the side of conservativeness. Our patients were stage 4 patients. We had 5 immunizations. Primary endpoint was safety, of course, and toxicity. And secondary endpoints were in terms of immune responses. The most important part of this, so typical protocol exclusion inclusion criteria, was actually a DTH response to recall antigen. And I think this is important so that you know that your subject actually has an intact immune system. So what did we see? We had immunized 6 patients. We saw, so in a 3+3 design, if something happens, you see some toxicities, you expand out to up to maybe nine subjects per dose, right? So if you see no toxicities, you actually end up with 6 subjects at the end of the day. So we had 2 doses, so 300 and 500. We really see no statistical difference between the 500 and the 300 dose. It's easier for us to actually use the 500 dose. Some of our gals are the IgG response, which is in panel A, have gone out for over a year now in some of them. So we're now thinking about writing a separate protocol to boost those particular gals. The IgM response, everything peaks at week 7. The IgM response peaks at week 7. The IgG response also peaks at week 7. Just like we saw in the mouse, we saw particular fold increases for the anti-GD2 titers, okay, in these particular individuals. And again, anti-GD2 is just being used as a target antigen. It doesn't necessarily mean that breast cells express GD2. Some do, and in fact, there's people who have reported that stem cells, breast cancer stem cells, part of their stemness is really because of GD2 expression. So that's something we're thinking about. This is a kind of an interesting story. So HCC 1954 cells are de novo resistant to Herceptin or trastuzumab. And we had looked at whether or not we can inhibit the migration, just like we saw with the human antibodies that we isolated with the column. We asked whether or not we can inhibit the migration of these particular cell lines. And as you can see that we do post-immune at week 7. More interestingly, we can kill these cells. So just by looking at MTT proliferation type assays, whether 231 cells, which are typically triple negative, HCC 1954 cells, which are typically de novo resistant to Herceptin, somehow we bypass whatever the resistance part of that is, and we're able to target that and kill it. It is through apoptosis. We can look at annexin. More importantly, we look at both the activation of caspase-3 and the inhibition of caspase-3, which is figure 3 on this side. So these are another types of cell lines, and this is kind of important for us because it also turns out that we don't kill MCF7 cells, right, which are typically caspase-3 deficient. So these are ZR75-1 cells. So we're able to kill cells that are caspase-3 functional. We cannot kill cells that are caspase-3 deficient. And that becomes important. In terms of the overall survival, just in passing, our vaccinees, so far they're at the 928 days for medium versus 312 days for the unvaccinated consenting individuals. So we are prolonging survival in these patients, even though it wasn't designed for a survival trial. We started thinking about combination therapy. We took serum from one of our subjects. We intermixed that with docetaxel. We could see that we can change the drug concentrations with the combination approach. So we changed the IC50 values with docetaxel with a fixed concentration of antibody. What was important for that, this lab study, was we went back to that subject. And she was typically given 2 types of drugs. For, part of our protocol was not changing her medication for 2 months prior to study eligibility. She had Mets. We decided to switch her to docetaxel, trastuzumab. We came back with a PET scan and noticed that indeed we were getting rid of her tumors, that at the end of the day and see that there was a baseline reversion of the metastases that we had seen in B over in this side. More importantly, this gal had brain mets. And as it turns out, we were able to, we don't see them anymore. So we don't want to say necessarily that we were able to get rid of her brain mets. However, they're not there. And as Yvonne pointed out before, sometimes T cells, maybe antibodies, and K cells are able to pass through what we perceive as a blood-brain barrier. In cancer patients, if people forget, there's a long history, long literature that there are leakages, in the blood-brain barrier of those patients, and consequently things can move. So we see this as an N of 1 in this particular story. But we're kind of excited about this. So we moved on to a phase 2. As a snippet, the FDA has told us for phase 3 we had a grade 3 adverse response cutoff, right? We didn't see anything in there. But for these patients in phase 2, we're targeting luminal A patients. So these are ER positive. The FDA came back and said we only want to see an adverse response 2 level, in those individuals. So the way we immunize is on the belly. This is a selfie from one of our subjects. All of a sudden, she started to balloon out in the 4 immunizations. She had 2 immunizations, 2 different spots. She started developing these types of, not lesions, but extensions, and consequently, we took her off the protocol. However, when the clinicians went back to look at her tumor, this is thinking about TCR as an endpoint. So as part of the trial, there's going to be 61 patients that are enrolled in this trial. This was actually our patient number 3. And consequently, her tumor has dropped about 90%. She was very upset, that she came off the trial. Her 10-week, but we had to take her off. Her 10-week immune response, though, is very similar to our other gals, in terms of her antibody response. So what we're seeing here is a very robust response in this particular person. And we think, somehow we are shrinking her tumors. Okay, so where are we going clinically? We're looking at a variety of other trials. We have a lung trial with our vaccine starting next week. More translationally, how are these things really processed? I mean, I'm still kind of mesmerized by the B cell compartments that the way an antigen is seen by B cells because of the carbohydrate nature. So, carbohydrates like to interact with B cells. How are they processed? And sometimes the kinds of responses we see really may depend whether it's an MZB B cell, B1, B2. So these are things that people don't really pay too much attention to. We do see a role for NK cells. We haven't talked about that. And we do have a role for T cells, which I didn't talk about, but gives us an idea of new peptide designs. Okay, so over the years, we've done a lot of different things. We had a lot of collaborators from Penn, a variety of other places, because we're also interested in bacteria and HIV. Funding was the NIH, DOD. Collaborators, a bunch of MDs, clinicians, and post and present members of our lab. So with that, I'll thank you for your attention.